Hello, everyone. Good afternoon, and welcome to our EMBRC technology webinars. With this webinar, we are closing our marine observation series, and we want to warmly welcome all the people that registered to this webinar from several countries, like in Africa, people from Turkey, from India, from Indonesia, and of course, our community from all over Europe. While people are still joining, I would like to briefly present our organization for those who are new to our EMBRC community. So the European Marine Biological Resource Center, EMBRC, is a research infrastructure that connects more than 80 marine stations, universities, and research institutes across 10 countries in Europe. I invite you to check our new website. Here you can browse our catalog, which includes more than 500 technology platforms, marine organisms, data workflows to support users coming from private sector and also from academia. One of our landmark initiatives is our Marine Genomics Observatory, EMO BON, which we are going to speak today. In this observatory, we follow changes in biodiversity all through the EU coastline, from the Arctic to the Red Sea. And what is unique of this observatory is that we do periodic sampling of water columns and hard substrates using the exact same procedures to sample, to process that sample, and to analyze the data. This ensures the high quality of the data that we extract from this observatory and makes our data comparable and reusable. We operate using the FAIR principles, and if you want to download the protocols that are used in our Marine Genomics Observatory, they are available in the UN Ocean Repository for Best Practices, and we will put the link in our chat. So through this initiative, we assemble a solid network of experts in ecology and marine observation. And this is why we want to spotlight with this webinar our colleague, Justine Pagnier. She's a conservation ecologist originally from France, with an academic journey that has taken her across Europe, primarily Sweden and Portugal. Since joining EMO BON more than 2 years ago, Justine has contributed to advancing omics-based biodiversity monitoring in Europe. First, she worked on tool development and data management, but today she is focusing in using all the data sets that she has produced for ecological research. Her current focus is on the detection and forecasting of non-indigenous species. She's using eDNA metabarcoding and biodiversity modeling approaches, which she will be sharing with us today. And if you want to know more about Justine's work, we encourage you to read her latest publication, which we will put in the chat. Before giving the floor to Justine, I would like to remind our audience to use our Q&A section, where you can type down your questions while Justine is presenting, and also you can vote for the question that you like the most. So without more introduction, I give the floor to Justine. Thank you, Justine, for joining us today.
Yes, thank you very much, Mary, for this introduction and also for the opportunity to speak today for this webinar. So I will start by sharing my screen. Sorry. Can you see my screen well?
It's coming, it's coming. Yes.
Okay, perfect. So we can start. As you probably saw, the topic of today is going to be environmental monitoring and identification of alien species. So Mary already introduced me pretty well. So you could understand that I'm initially a biologist. I did conservation biology, more like all sorts of habitats and ecosystems, but progressively I focused on the marine side of things. And in last November, I started my PhD in Gothenburg University, and I will, during this presentation, talk about the work that I've been doing this past month. And I will show you a brief outline of the talk of today. So we will start by a bit of context in which that presentation is coming. So then we will go through a few monitoring tools that are being used today to monitor marine biodiversity. I will then introduce you to EMO BON network. And then we'll talk about potential uses of genetic data and computer vision, mostly in the team, in our team in Gothenburg University. So first, the main question is why do we want or why do we need to observe the ocean and to understand marine biodiversity? So you probably all know here today that the ocean is highly threatened, highly disturbed at the moment. So we need more and more biodiversity data to understand how the natural ecosystems are reacting to those disturbances, to make better decisions for conservation. For example, now there is this new concept of essential biodiversity variables. So a lot of parameters, sort of, that we want to understand better about marine ecosystems. And then we can see how they react to climate change and how we can interact better with marine ecosystems and how we can use them in a more sustainable way. And all of this is connected to the One Health concept that probably some of you have heard about, which states that everything is sort of connected: human health, environmental health, and animal health. But the point also with ocean observation is that monitoring marine environments is quite challenging because the ocean is very vast. It's also highly variable, meaning that different areas of the oceans might behave differently. So you cannot apply one thing for all kinds of areas. And it's also quite hard to access and to monitor. For example, if we talk about the deep sea, it's really hard to reach there and to go observe the life in the deep sea, or some areas of the open ocean, or maybe the sea around the poles. It's not always easy to go there. So all of these questions that I just talked about, they fall into this Ocean Decade Initiative, which is led by the United Nations. And it started in 2021, but it goes until 2030. And it's basically composed of 10 different challenges that are all about understanding better marine ecosystems, acting in a more sustainable ways with them. So there's a lot of work to do with those challenges. And of course, we are already halfway through that decade. So it's still a lot of work to do before 2030. But of course, those topics will keep being the focus even after that date. Challenge 8, for example, is about creating a digital representation of the ocean. So in Europe, we have the European digital twin of the ocean. So the idea here is to make use of all the data that has been collected for years now, to put all of this, integrate all of this data together and combine it with tools like models and AI and a strong infrastructure to build basically a big model, a big representation of the ocean. And this tool, in the end, can be used by all sorts of people, not only people from academia, but also people from the industry or decision makers. And the idea is to do what-if scenarios. So what if we do that kind of, action towards the ocean? What's going to happen in the future? How is that going to impact the ocean? And the novelty here is that it's an iterative method. It's not only about doing one model, seeing the output, and making a decision. It's about having constant data flow into that digital twin of the ocean so that it keeps learning and it keeps checking how the ocean is doing. So now we can talk about monitoring tools. So for a very long time, the way to monitor the ocean was quite costly and labor intensive. So you had to go out there, usually send divers if you needed to go pretty deep. So it was, very intense. And also you had to count and identify species manually. So it needed a lot of taxonomic expertise. And as I said, sometimes it was hard to access some really remote or deep environments, So all of this created some sort of spatial and temporal gap in the data. And it was also really tricky to standardize methods across larger regions. But with time, so here you have the case of Sweden, for example. With time, researchers could go further. For example, if you see the picture on the bottom left corner, it's Kristineberg, one of the marine stations we have on the Swedish west coast. And at that time, maybe 100 years ago, people were usually monitoring ecosystems pretty close to the station, a few hundred meters or a few kilometers maximum. But now you have big boats or even robots that can go by themselves out there in the sea and take samples, take measurements with a high level of precision. So all of this created kind of the rise of data-driven ecology. And what does that mean? It means that we are basically flooded with data now compared to a few years ago. We have a lot of genetic, remote sensing data or citizen science data and also image data. So the new questions now are how to efficiently process all of this data and how to use it correctly to get information from them. So 2 methods I want to talk about. Probably many of you today know what's environmental DNA, but it's DNA that is collected from a variety of environmental samples. So it means that it's not directly obtained from the organism itself, but more like traces that organisms left behind them in the soil, in the seawater, snow or ice, air, gut content, and feces. So this method is very powerful because it's quite sensitive, and also it allows you to detect species that are smaller and you couldn't really see them with the naked eye, or they are more rare. So if you go out there, you don't really see them, or some more cryptic species that are hard to identify. Environmental DNA is pretty powerful for this. And another tool that is used now to monitor the ocean is autonomous reef monitoring structures, or ARMS. So they are stacks of 9 PVC plates, not very big, and they act as a kind of hotel for species that live on the seafloor on hard bottoms, because they are deployed for a few months on the bottom of the sea. And basically things will go there, will start to settle there on those plates and grow. And after a few months, the stack of plate is retrieved. And everything that grew on top of it, and also the water around, everything is sampled. So you get basically a mix of organismal and environmental DNA. And you can get DNA out of it, and then a list of species that were around that arm structure. And as I mentioned, it's a technique that is used all around the world. So at the moment, you have more than 2,500 arms that were deployed around the world. As you can see, it's not very even, like some regions are more favored than others, but that's usually the case when monitoring marine biodiversity. And on the other graph, you can see that those arm structures, they can be used to detect a wide variety of taxa. So it's a very good sign. They can be used for different purposes. Another thing that I will talk about mostly towards the end of this presentation is image analysis, and mostly through computer vision. So for example, here, this video, it's a few meters deep, so a place that is not really easy to reach. And as you can see, there is a lot of things going on. So what was happening before is that people were still taking videos, but they had to then sit in front of their computers for hours and count and identify everything manually. While here, they just trained a model, and the model is doing the work for them. But I will come back later to it. Okay, now we can talk about EMO BON. So Mary already introduced it briefly, but basically EMO BON, which is the European Marine Omics Biodiversity Observation Network, is a large-scale project that is led by EMBRC, and it has started in 2021. And the point here is to collect DNA samples in different places of Europe, and different kinds of samples. So we want to get a holistic view of the marine habitats that exist in Europe. So,
excuse me, Justine.
Yes.
We are still seeing the monitoring video in your slides.
Maybe it's...
There's a delay, so maybe if you can restart the slide.
Okay. Yeah, I will.
Thank you.
Do you see it now?
No.
Wait, maybe...
Did you stop sharing?
No, I didn't stop sharing, but I can maybe stop it and restart it.
That would be best.
Yes, okay. Okay, let me know if you see it, when you see it.
Okay, it's charging. It's all black. No,
Maybe
it's black.
yeah, I will.
Yeah, I should try to share my screen then.
Sure, yeah, if you think it's better.
Yes, because it's still black.
Yeah.
Well, now you stop it. So let me share from my side. Okay, no. So we're going to start from this one, I guess. Yeah, there you go.
Okay, thank you. So, yes, as I was saying, EMO BON was launched in 2021. Yeah, I was saying that we try to get a more holistic approach here and sample different kinds of habitats. So the soft substrate, the hard substrate, and the water column substrate. So, Mary, you can change the slide now. Yeah. So if we start with the hard bottom monitoring, so that's where we use the arms structure. And all of those red dots are observatories where those arm structures are being deployed, usually once a year. Then it always depends what people are able to do. But the point is to deploy those arms on a regular basis and retrieve them on a regular basis as well. As you can see, ARMS EMBRC, so the network of ARMS, started in 2018, so it predates EMO BON. And to this date, we have more than 20 observatories across Europe. Yeah. So that's how they look like. You can see that in different areas of the world, they might look very different. And that's kind of the beauty of it. It's that with a standardized method, we can still compare different areas that Indian look very different, and the communities are very different depending on where you are in the world. The water sampling now, so here the focus is mostly on plankton. So people are going to sample once every 2 months during daytime, and they use this kind of long filters. The filter size is standardized, again, to sample the same kind of organisms. Then there are options to focus on larger planktons or also on bacteria. So there are flexible protocols also here. And finally, the sediment sampling. So here the focus is on meobenthic organisms, so creatures that live on the sand and the mud in the seabed, on the seabed. And here the way it works is that people are using 10 centimeters wide sediment cores at low tide. And yeah, it's also possible to sample like microbial communities or microbentos, so like larger individuals. So as I mentioned, the point also with those kind of big European networks is to have standardized methods. So not only on the field, but also when you go to the lab then to sequence the DNA. So what happens is that partners send their samples to a centralized sequencing facility, which is Genoscope in France. And there, the people are going to extract the DNA from the samples and perform the sequencing. And what comes next is that we really care in those networks about making the data fair, which means findable, accessible, interoperable, and reusable. So we share the raw data, so the sequence data, But also in those arms data release, you can find the process data, which is basically then put into Darwin Core archives, which means you will have the species identification and their presence and absence across the data set. So those data release, which are like data papers, basically, they explain everything, how the data was collected on the field, processed in the lab, and then processed in terms of bioinformatics. And it's a very good tool for anyone to then come to the data set and use this data for their own scientific questions or own projects. The point here is that even people outside of the network, they can just access this data and use it for their own projects. So we have 3 working groups, science working groups at the moment in EMO BON. The first one is working on marine microorganisms, which are all of those little creatures that you cannot see with the naked eye, but that are still very important for the ecosystem health. Then the second working group is the benthos, which is all of those creatures living on the seabed. And the science working group 3 is about non-indigenous species, and I will come back to it very soon. So one of the first observations that we got after the first sampling campaign, for example, of arms. So on the left side, you have the first sampling campaign, and on the right side, you have the second sampling campaign. And here the observation was that arms are pretty good at detecting biodiversity, measuring biodiversity, because we got good signals from the main phyla and the main species across Europe. So this is already a very valuable data set for biodiversity monitoring. And then we also realized that through this data set, we could detect ecological key species or indicator species, for example, those species that are very picky in the places where they live because they are very sensitive to disturbance. They are the ones that are used as indicators because they will react first when something bad happens in the ecosystem, so they are very important. We could also detect alien species and endangered species. Yeah, so now I will go through one case study with you, which is this paper that we published in February, and it's also the first chapter of my PhD thesis, and it's about alien species across Europe detected with ARMS EMBRC data. So maybe some of you here know that alien species are one of the top 4 greatest threats to the world's ocean today. They are, as you can see, since the 50s, they increased drastically in Europe. And those are species that can be moved to a new place, for example, with shipping or some deliberate release, or they can also naturally move to a new place because of maybe climate change, for example. And some of those species can be perfectly fine in the new area where they are, but some other species can suddenly create strong damage in the ecosystem. So that's when they are called invasive. And that's why rapid identification and management are crucial. So what we did in this study is that we used data from 112 ARMS units. So that represent arms that were deployed across 19 observatories in 14 countries and between 2018 and 2021. So I'm not going to go into all the details of the analytics here, but if you are interested, you can scan that QR code down there where we published all of the bioinformatics pipeline, basically. Also, we just the main conclusion here was that we detected 63 non-indigenous species across the whole study area, so across the whole Europe. And they were mainly arthropods, mollusks, bryozoan, and cortalids. We could also detect, our most widely distributed alien species in these data sets were already famous aliens in Europe. So it was a good sign that in this data set, we could also detect them in many observatories in a way. What we did as well was building maps of which observatories, ARMS observatories, had more aliens than others. So the first map, the A, is made with one genetic marker, and the other one is another genetic marker. So they cover different kinds of taxa. And you could see, for example, that Ravenna in Italy, the point that is in the north of the Adriatic Sea, this was kind of a hotspot, or at least an observatory that had more aliens than the others. Same thing with Plymouth in the English Channel, had quite a few aliens. The last thing that we found with this paper is that even though many alien species that we detected were already known in the places where we detected them, some of them were new to the area where we detected them. For example, that species in the Adriatic Sea and two others in the Red Sea. So those cases are the important ones, or like the ones that can be used for the early warning systems, because if we detected them, maybe it's an artifact, but maybe it's a sign of early detection or early settling. So you want to be able to go back to the field and check if those species are really there. And then you want to be able to tell if they will impact negatively the area or not, and if you need to act fast, basically. And this part now is what I'm working on at the moment. So once you get all of those occurrence data from alien species, you can do suitable habitats modeling. So try to infer which habitats in Europe are suitable for species A, for example, or here Bonmesonia amifera, which is an alien algae. And you can do this for today's scenario, but also in the future, for example, here in 2100. And under different climate scenarios. So the green one, SSP 126, for example, is about what would happen in terms of climate if we do better than we are doing now, basically, if we reduce fossil fuel industries or that kind of things. The orange one is if we keep doing business as usual, and the red one is if things don't go as well and we go ham on fossil fuel industries or that kind of technologies. And the goal after is to kind of stack all of those individual maps to identify areas that are more vulnerable to invasions overall, or at least over the whole of Europe, to then build this early warning system. You know that if, for example, here we see that the North Sea, the English Channel is pretty red, so maybe that's the area where a lot of focus for alien species should be put, for example. And then we can overlap this with major ports and see if there is a correlation with the big ports and the highways for ships, for example, or if there is a correlation as well with the MPAs. And the final thing that we want to do with alien species is classify them, because as I mentioned, some of them are hitchhikers, so meaning they will be transported by ship over a long distance and suddenly arrive very far to their native, from their native range. Some of them are range shifters, which means that they will move slower, usually with climate change. For example, they will move northwards. Some of them are more cryptic. We don't really know how they move or what's happening with them. But we build a tool which is called Alien Detective. So the way it works is that it measures all the distances between our arms locations, so where we detected species A, and then all of the other GBIF records. So GBIF is the Global Biodiversity Information Facility. So it's a big database that records many occurrence points for many marine species, or many species in general. And then by having all of those distances, you can plot them and see the frequency of each distance, of the distances for each species. And by seeing the patterns of those plots, you can kind of classify the alien species into those different categories. And this is very informative also for invasive management. So for example, you have the first graph, it's a species, or like the question is which species are newly introduced in a specific location. So for example, the Gulf of Piran in Slovenia, you can see that most of the species were, like the GBIF occurrences are pretty far from the Gulf of Piran. So those species are probably new to the area. Or the second graph, which location has a specific species been introduced. So for example, that species probably was introduced in the Gulf of Piran quite recently. Okay, so now we can move to from genetic data to image data. And I will mostly focus on what we've been doing in Sweden, or mostly my colleagues, because I'm working mainly with DNA, but we have a lot of colleagues in our team working with the SubSim, so the Swedish National Platform for Analysis of Subsea Images. So I will go through a few examples of what they've been doing. For example, here, the seagrass habitat mapping close to offshore wind farms, because there is this hypothesis that offshore wind farms, they can kind of offer a certain shelter for these plants and also for macroalgae. And if you have a lot of seagrass and macroalgae, it's basically building habitats for other species, for example, invertebrates or fish, because they will find shelter there. They will have their nursery there and they will feed there. So it's usually a very good sign for the environment. So we had a student that recorded a lot of videos and then trained an algorithm, like a model, to detect the coverage in seagrass, in algae and sand, and kind of calculate the coverage. Another example of image data analysis is for alien species monitoring. If you see, you have the black goby in pink in the video, which is the native species, and you have the round goby on the background in orange. I hope all of you can see and can read, but basically you have one native fish and one alien fish that look very similar. The alien fish, sorry, has a little dot on its fin, so that's how you can recognize it. And people used to take videos and check those videos manually, but now, again, you can train models to automatically identify the species and see, for example, how long do they stay in the frame and how do they compete with the other native species. Another example, so this was about monitoring benthic biodiversity in aquaculture. So to kind of estimate the impact of aquaculture on benthic communities, especially here, fish and some big invertebrates. Or here another example, monitoring key species for fishing, for example, the European lobster. So in protected areas or in fished areas, and see if there are any difference between those two types of areas. And also here, what was done was measuring the size, because it's very important for some species, the size distribution. And usually people were monitoring these species by having them basically trapped in these traps, but they realized with this method that there were some bigger individuals that were never caught by those traps. So here, computer vision was pretty useful in that sense. And the final example, which is monitoring effects of management plans. So here I will talk about the case of the Koster National Park, which is a marine protected area on the Swedish west coast that was inaugurated in 2009. Yeah, can you click a second time, Mary? Sorry. Yeah, because the videos. So the good thing here was we had videos from 1999 that were recorded in the area. And we had Christian, a student here in our team, that basically trained the model to recognize like sort of iconic species for that area, and then apply that model for all of those videos. So I only put 3 dates, but we basically had many date coverage from 1999 up to 2023. And so for all of those species, there were different patterns, but I will, for example, show this one. So those two species, they really dislike when sediment is falling on them. And this is usually a side effect of trawling. Trawling is creating a lot of sediment in the water column, and then this falls back on those species, and they really dislike this. And we can see that from the inauguration of the national park, their abundance kind of increased drastically. So the way you read those graphs is depth on the y-axis, and on the x-axis, it's an approximation of the abundance. So this tool was really good to kind of see a real impact of an MPA implementation on the ecosystems. So as you probably understood until now, observation data is really informative and really essential actually to take better decisions. For example, when we talk about marine spatial planning or blue economy, it's very important to, of course, focus on the economy and the feasibility, but we also need to ensure that those decisions, they make sense in terms of sustainability. Are they really useful for the ecosystems? And the only way we can be sure of that and we can know that is if we have good observation data and especially recurrent data. We have to monitor constantly the ocean to see how it reacts to our decision. Then if we talk about marine protected areas, maybe some of you know that we are supposed to protect 30% of the ocean by 2030, which is in 5 years from now. So we have to act fast, but we also have to act smart, which means that we have to ask ourselves which 30% do we want to protect. And there are probably areas that are more vulnerable or areas that are playing a very important role for the ocean health. And then those areas would be the areas to protect. And only through observation data we can know this. Same thing for the EU Marine Strategy Framework Directive. We want to achieve a good environmental status overall, and this needs observation data again. Which means that what's next for the ocean monitoring overall? So I think with time we will have an even better real-time and in situ technologies that will be able to give us precise data in a very efficient way and almost on a real-time basis. And this will also be done through the integration of those data streams, so remote sensing data, genetic image data, and also citizen science. So all of this kind of data formats and data streams should be integrated together to be more informative. And this will also create a better coverage and representation of different kinds of habitats. And hopefully the main point is also to have a good link between science and policy. So we need to know exactly what policymakers need to understand, to be informed on, so that we can provide as scientists a good translation of our observation data. So yeah, this is a few references I used for my presentation. And thank you very much for listening.
Thank you very much, Justine. So while all our participants start sharing the questions in the Q&A section, I will read the two questions that we have already there. So first of all, I lost it. Oh, it's here. I would like to, from Youssef Afquir, I would like to ask if you have any knowledge or experience related to the mapping of macroalgae and their ecology. I have just graduated with a master's degree in environmental science and marine biology, and I intend to continue my studies with a PhD in the same field. So any resources, guidance, or advice?
So yeah, I started working on mapping and especially modeling species habitat suitability, and only recently when I started my PhD, but I found this Biomod 2 package, which is very good to integrate data from different sources, so occurrence data and environmental data from different sources, and then combining it together to kind of map any species of interest, really. About their ecology, it's true that then a lot of factors should be taken into account when you want to map the distribution of a species. For now, the way I've been doing it is just correlation between occurrence and environmental variables, not really, for example, when you talk about competition or like interspecies interactions. This I'm not working with at the moment. But yeah, that would be from my experience working with Biomod. And there are a lot of data out there at the moment, a lot of data that you can freely use. BioOracle for environmental variables, for example, is one of the data source that is very used, or EMODNet. Yeah, then it depends on what you want to do exactly, I guess.
Thank you, Justine. We have another question from Gilbert Romero Suarez, which is a comment more than a question. He comments that morphological classification of organisms is still necessary. eDNA techniques are complementary to morphological classification. Not a single answer is 100% correct. They complement each other. What is your opinion, Justine?
Yeah, definitely. I think during this presentation, I presented a lot of new emerging monitoring techniques, but they really work hand in hand with the so-called more traditional methods. Well, I think it's still very important to get to keep a taxonomic expertise in that field to really understand what we are working on, especially because I think that the risk in the end would be to only work with DNA sequences and not really be connecting with what those sequences actually mean. So always linking the DNA to the morphology and then the functionality. There are a lot of answers in the DNA itself, but not all of the answers, of course. And eDNA is not flawless either. So you have a lot of limitations still at the moment. So of course we work to kind of reduce the impact of those limitations, but by having some sort of integration between eDNA and, for example, you mentioned morphology. So a lot of people are working on bigger tools to kind of merge the two, connect just the morphology of one individual to its DNA. So yeah, there is still a lot of work to do on that field.
Thank you. I was wondering if there is any collaboration foreseen in EMO BON with policymakers, for example, in order for your work and your conclusions to be taken into account for marine special planning, for example.
So that's for sure one big goal of EMO BON is to transfer the science to the policy making. I'm not sure of all the details of what's going on at the moment, but a big desire of EMO BON is to get answers from the policymakers. What do they need? In which format do they want the data? Like, because scientists usually work in a certain way. You know, for example, Darwin Core Archive is a very famous format for presence, absence of species. So we know how to work with this, but maybe they need other kinds of tools to kind of answer the science that we do. So there is a lot of communication. There are lots of, let's say, meetings between different organizations across Europe to just first understand each other. What do we need and what do we want? And then translate the science.
Thank you very much. You mentioned Alien Detective. So this is a software or a platform, I understood. Where we can find it? Can you maybe share with us the link or?
Yeah, actually, I had updated my presentation for this, but you still had the previous version. But yeah, so it's a tool that we've been working on for a few months now. It's still in the testing phase, but the point is at some point to release it. So the code is on GitHub, but we are still trying to scale it up to have a lot of species that can be processed with that tool. And yeah, it's gonna at some point be published probably through a paper and an official release of that tool.
Okay, great.
Yeah.
Well, we are all very excited waiting for Alien Detective to come out. But yeah, I had another question because we don't have for the moment, so I invite all the participants if you have other questions for Justine. And in the meantime, about the behavior from the videos that you show, can we extract conclusions then for the behavior? For example, I don't know if there's some kind of competition or aggressive behavior against like the local species. Is this
Yeah.
what can be done?
Yeah, it can definitely be done. So again, I was not the one working extensively with those videos, but I know that you can kind of first count the number. For example, that video with the two fish, the native one and the invasive one, there is food in the middle. So you can kind of see if they compete for it, if one is pushing the other one away. Like, you can definitely do behavioral analysis on those videos. Those videos are actually like very valuable resources, but I'm not sure how it would be translated in terms of building a model for it, because right now what models are really good for is just identifying and counting and deriving abundance from it. But you can definitely look at those videos for other purposes, for example, behavioral analysis.
Okay, great. And finally, Gilbert Romero Suarez has another question. When it comes to detecting invertebrates, how good are the solutions that you have presented? Are there some species that are more difficult to detect than others?
Okay, so yeah, if we talk about eDNA, so the strength of eDNA is that it's highly sensitive. So even smaller invertebrates or invertebrates that you would really struggle to see if you would just go dive there, it might be detected by eDNA. So that's for the strength part. However, the biggest limitation of eDNA is that you need a reference database. So you need to already know the sequence of the species to then match it. And that's why, again, traditional taxonomy is very important. But yeah, basically, you sometimes will have some species that will not get matched to any species name. So maybe some invertebrates in that sense are being overlooked, because invertebrates, it's like a group of species that is still a bit understudied compared to, for example, fish or big mammals that we know a bit better. There are many invertebrates that we probably don't know about at the moment, so they wouldn't be detected by eDNA. And in terms of computer vision, it's mostly about what you can see and what you can identify. So in terms of what you can see, it's how good the quality of your video is and how precise it is, and a lot of things will come into play when it comes to the ability of the model to identify a species. Like, you need good quality, and if it's a small invertebrate, it might struggle more. And especially when we talk about invertebrates, you have a lot of species that look very, very similar but are not the same species, and that's when genetics is usually required. So the big invertebrates, let's say, for example, I talked about the lobsters or some crabs, might work really well, but like the smaller ones, it would be limited. But I know that some people, so not in our team and not from projects that I know really well, but some people work on like applying those object detection models on like microscopic videos, and then it's also a new challenge, a whole new challenge.
Thank you, Justine. And we have one last question before we launch a poll for our participants. And it is a question from Matteo Bolsonella. You show data coming from several campaigns and technologies. Were the data coherent in terms of units, or did you need to post-process them? How did you cope with the uncertainty associated to the data and methods used?
Yeah, so here I think I can talk mostly about this paper on alien species, because we used a big part, like a big majority of the arms data. And no, it was not uniform in terms of, for example, arms units deployed in different observatories. Some of them had like 3 deployments, some of them only 1. Same thing with the deployment duration. Some of them were only like 4 to 6 months, and some of them were 2 years. So, for example, in those 2 maps that I mentioned, that I showed for the geographic distribution of alien species, we had different bars of different colors because we had to create sub data sets, kind of like locations or arms that had similar duration, similar number of arms, and similar, more like kind of normalize the methods across the whole data set. Because this paper was done with data that came from like 2018, which was the beginning of the project. So there was a lot of testing. So that's why, yeah, it was less standardized than what we are aiming for. We really aim for something that we can easily compare across across areas and across time as well, have this kind of time analysis, time scale.
Thank you, Justine. We are very happy to have you here in our tech webinars. It was super interesting. And we hope to have you again in the future. And I would like to launch a small poll for all our participants, because we would like to shuffle a little bit the format of our tech webinars. And we would like to know your opinion about the format of having one speaker, or if you would like to have like roundtables, and also which kind of topics you will be interested to hear about. So if you could give us your opinions, they will be very well received by us. Thank you. While people are responding, I want to just remind that Justine's webinar and all our tech webinars are available in our YouTube channel. So you can follow them and watch them again how many times you want, and also that our next series will be by the autumn this year, and we will be having bioprospecting. Will be the topic of our next tech webinar series. So, well, thank you very much, everyone, for joining, and thank you again, Justine, for being here. And I wish everyone a very nice afternoon. Take care.
Thank you very much. Thank you for the opportunity, and thank you everyone for coming today.
Thank you. Bye.
Bye.